Buenos dias, señoras y señor. Greetings, everybody. I'm Geoffrey Eglinton, and I'm speaking to you from Bristol. And I'm really sorry not to be with you at the conference, but your steering committee has kindly asked me to start the first plenary session with a talk on the expedition we made way back in 1960 to Tenerife in search of leaf waxes. Now, you may wonder how on earth this came about, and I must admit I have too. And so over the last few weeks, actually, I've been pestering old colleagues and trying to find out just what sequence of events took place that resulted in us chasing plants all over the island here with a gas chromatograph in tow. Now, it turns out that this story is really not a logical one, and I've often wondered just how science gets done, and I'm quite convinced most of it happens as a result of accidents and chance meetings and looking in the wrong book or reading the wrong paper. And I think the story I'm going to tell you illustrates that. First question was, who is it who went on this expedition? The second question is really why, which is quite hard to answer as it turns out. And then later on, one can look at what actually we got out of it, and of course what it has led to. And very briefly what it's led to is a very large field of work which now uses the geology and geochemistry of leaf waxes. Who went was actually, of course, myself, a research student, one wife, two small children, a grandma, a gas chromatograph, and an infrared machine, and a few other things besides. So this strange conglomeration went to Tenerife in 1960. But you might ask, why me? I was nominally what's called a pure organic chemist, but already by that date, I was beginning to metamorphose into what we now call a biogeochemist, although I wasn't aware of the term at the time. And this took place because I was involved in analytical instruments, and I managed to get hold of a brand new instrument called a pi-argon GC, which was incredibly sensitive for its time and really opened up a whole new lot of science that was possible. This amazing gas chromatograph was called the pi-argon GC, and it had been invented by James Lovelock, who of course is well known for his Gaia hypothesis. And Jim Lovelock had told me about this instrument, and we managed to get it at Glasgow. Now, it was so sensitive for its time that one could do a lot of things to push it into more difficult tasks than the usual contraptions. So we put it up to its highest temperature, we increased the gas flow rates, we reduced the stationary phase, and so on, and we found we were able to get quite polar molecules and big molecules through it. And so we decided this was a new tool, really, for analyzing complex mixtures of natural compounds. So we put out a note in Chemistry and Industry in 1959, and this resulted in about 1,000 reprint requests coming in the mail to Glasgow. Now this is, you might think, well, why weren't they using Xerox? Well, of course, Xerox wasn't invented at this point, and copying things was really quite a trouble. So people wrote in for reprints of articles. Well, why on earth did we get bombarded with all of this? Well, it was because we had made a list of all the things we found we could analyze, and we published that in the paper. And you can see that in this list here. Now you'll notice that a C26 alkane came out in about half an hour. but a C36 alkane took about 5 hours to come out. So you can see that we were in the range round about C30, which we could analyze in a reasonable time. So the question then is, what should we be looking at? The way really seemed open to analyze all sorts of complex mixtures which were known in nature, such as essential oils, perfumes, fats, and of course waxes. Now, the waxes are the key thing for the rest of this story, but this was quite a challenge. With this great possibility of doing all sorts of analyses, what really should we concentrate on and try to find something really exciting to look at? Well, to some extent, the answer came in the mail, because someone who had read our article and seen the word waxes in it was Professor Chibnall, who was a very distinguished biochemist at the University of Cambridge. And he wrote and said, look, would you like our collection of plant waxes which we've accumulated through a lifetime of research? So of course I went down to Cambridge and was ushered into the great man's room in his home and presented with a huge pile of W.D. and H.O. Wills metal containers that had all been full of cigarettes, of course, at one time, and they were now full of lots of small glass tubes, beautifully labelled, containing the results of his work of many years. Well, at Glasgow we investigated quite a few different sort of interesting mixtures, but eventually I came across the work of Holger Erdmann, who is a Swedish chemist, who had shown that it was possible to classify organisms to some extent using the compounds they contained. This is an area called chemotaxonomy. And Holger had, of course, concentrated on things like phenols and terpenoids of various types. But I wondered, what about these leaf waxes? Surely they might be interesting. So I looked into this a bit and came to an interesting conclusion. What we realized was that with the pi argon we could now analyze these leaf waxes in some sort of reasonable time. And the reason why the leaf waxes looked really interesting was because these waxes are on the outside of all leaves and protect the leaf in various ways from all sorts of things and control the water movement. So they are really in the front line of evolution. The plant has to conduct its war with the environment of all types using the leaf waxes on the outside. So they must be rather good from the chemotaxonomic point of view that Erdmann had looked at in terms of being the result of evolution of those plants. So what about then making this a real effort, a chemotaxonomic study of certain components of leaf waxes? Here I can show you a typical GC of the time in which the C31 alkane is the dominant one. The nice big peaks which come out in a reasonable time. So this was the basis of our proposed chemotaxonomy study. What about doing a Darwin, i.e. imitate Darwin's work on the finches of the Galapagos, but instead of using the beak shapes and size of his finches, use the distribution of these alkanes in the leaf waxes of a group of plants. And, well, for this, then, we would need an island, preferably, which was pretty isolated, fairly young, so the plants haven't gone completely wild, and we need to be able to find these plants and analyze them. So the Galapagos is an awful long way away and also doesn't have much growing on it, but the Tenerife, the island in the Atlantic, amongst the other Canary Islands, the Fortunate Islands, these looked a real possibility. Now you might ask, how did I know about Tenerife? Well, it comes about because we'd had a postdoc visiting in Glasgow who had been born on the island of Gomera, which is just off from Tenerife. And in fact, he'd done his PhD studies at the University of La Laguna on Tenerife. So he had told us about this wonderful area. So I thought, Canary Islands, Fortunate Islands, sounds good. So that's how we started with the idea of doing this Darwinian type expedition. Now, you might say, well, why not just go out and quickly collect the plants and bring them back? Well, that's true, but we would get a much more, I thought, sensible selection of plants by actually taking our gas chromatographs with us and analysing the things, and this would guide us as to what to do. And then that raises all sorts of questions as to, we need a place to do the analyses, we need colleagues, we need experts, and so I set about trying to solve these logistical problems. Now, the more I looked at the Canary Island idea, the more I liked it, because it turned out that Tenerife geologically was very, very young, about 10 million years or so, and still covered with large areas of volcanic rocks and very mountainous. And it also had already a population of waxy plants called the Crassulaceae, which of course had colonized the islands, being blown over, pollen and so on, from the mainland of Africa. So, and then of course it had this University of La Laguna, which sounded a very good base for the project. So we set about trying to make this come to fruition. We needed a scientific base, and we needed some Spanish colleagues. Now, it so happened at a dinner down in Cambridge, I accidentally found out that Professor Gonzalez, who was an organic natural products chemist at the University of La Laguna, was anxious to acquire gas chromatographic equipment. In discussions, I learned about Professor Gonzalez, who turned out to be a very distinguished natural product chemist who had set up a new laboratory on Tenerife at La Laguna. In fact, he went on to win numerous prizes, the Prince of Asturias Prize, for example, which has only recently been awarded to people like Jane Goodall and Venter, the DNA expert, and so on. So it's a very distinguished company. Anyway, he really was anxious to collaborate, and the addition of Dr. Sventenius completed the team. So it turned out then that we could really fit everything together very nicely by us taking out gas chromatographs, etc., and setting them up to work in his laboratory, and then he would actually buy them and would have a readily trained crew as a result of our visit. So everything fitted together. So we set off for Tenerife, but the plan now also involved a Dr. Sventenius, a distinguished botanist who was in charge of the botanic gardens at Puerto de la Cruz on Tenerife. So we had all the bits in place, except for leave from the university and some money. So the university gave me 5 months, and the Carnegie Foundation came up with funds. So we were now all set. So the whole expedition, wife, children, research student, grandma, etc., we all descended from the ferry boat on Tenerife in August 1960 and went up to the University of La Laguna. And it was a beautiful sight. and still largely undeveloped areas around of volcanic rock where bananas and other things were being cultivated. So Professor Gonzalez turned out to be really anxious to show us around his beautiful island, and he turned out to be the owner of a beautiful white jaguar, the only one on the island. So we went all over the place looking at various views and so on. But one thing that was noticeable, that everywhere things seemed to be growing well, they turned out to be banana plantations. So we had some difficulty in deciding how to find these Crassulaceae, the wild endemic flora that we were after. However, with the help of colleagues and particularly Dr. Sventenius, we got to these canyons and so on where they were. But we also went off and had a few interesting trips on our own, and one was an expedition up Pico Teide, and in those days there was no teleferique or anything, so you had to stagger the whole way up. And so here we are on the top in the dawn, with the shadow of the peak falling across the sea to the island of Gomera that I mentioned before. This crater on the top was a classic fumarole with lots of fumes of sulfur and so on. So this was quite an interesting thing to see, and I gather you're not allowed in there now. Anyway, the actual field expeditions took us into all sorts of canyons and little crevices that the farmers couldn't get at, and there we found these Crassulaceae and collected some 50 or so species, which we were then analysing back at the laboratory. So, at the end of the 5 months on Tenerife and the analyses in the laboratories at La Laguna, what did we find out? Well, we accumulated the histograms of some 50 or so species and looked at them very carefully. Well, we summarized the findings in a letter to Nature and a detailed report, phytochemistry. And these came out early in 1962. So I'll quote from a few of the conclusions for you here, and I will then comment on them. Even in the present limited investigation on a restricted area has shown at least some glimmerings of a taxonomic relevance in the leaf wax hydrocarbon pattern. Now, I think you will admit that agree that we are being fairly careful here. So some glimmerings is not sounding a very marvellous Darwinian type achievement, but it really was difficult to come up with something really dramatic in terms of evolution. But I think you can see some of the patterns do show similarities within the genera. So that's why we call it some glimmerings. And then we finished with such investigations would not only be of taxonomic interest, etc., but could be used to study relationships, plant waxes, and things like plant metabolism, water balance, plant diseases, weather damage, atmospheric pollution damage, efficacy of crop spraying, the carcinogenic effect of tobacco smoke, and the origin of petroleum. Now, ladies and gentlemen, I can't offer you any dramatic biomarker phrases or phrases about proxies for environment because we hadn't considered them. But we were very actively considering this origin of petroleum thing. And one thing that was quite dramatic about these results was that the N-alkane patterns were very similar to those found in recent sediments. And this brought us right into an argument with the petroleum people who had said, this is petroleum. And of course it wasn't. The hydrocarbons in recent sediments, in unpolluted, were quite different, but exactly parallel to the leaf wax thing. So we began to realize that really these leaf wax studies could make a contribution to the whole subject of the origin of petroleum. Now, you might be wondering, those of you who are not involved with plant waxes, just why are these things so important? And I take you back to 1959, when a Glasgow colleague once stopped me in the lab and said, "Why are you people working on N-alkanes? They don't do anything." And that's precisely the point. Their very inertness makes them valuable tools for exploring the past history of environments and so on. And of course, the whole modern work going on at present is based to a large extent on that inertness. So now, over 50 years on from 1960, where are we exactly with the leaf waxes? Well, one important thing is we know that they are global wanderers. They travel across the face of the globe and get deposited all over the place, particularly in ocean sediments, and so they provide us with a record of what's been going on the continents at that time. So that this history, of course, is recorded not only in the carbon number distributions of the homologs, but also in their isotopic contents of the carbon 12-13, carbon 14, and deuterium. Now, one thing that makes them very valuable here is that the result of aerial mixing, that's area in terms of geographic area, and with the wind systems, is that there is a sort of averaging of the data, and this provides you with a very broad overview, but nonetheless a valuable broad overview, for example, of the C3 versus C4 question. And this is illustrated in two posters from our colleagues. So as phytogeographic proxies, they reflect the climate, the atmospheric CO2, the regional environments, the particularly temperature, aridity, and the wind systems. Just looking back now on the expedition in 1960, it may not have actually provided a really dramatic finding at the time, but it certainly was part of my metamorphosis and has led me into lots of exciting research on leaf waxes in different parts of the world, particularly West Africa. But all I can say is that if I look in the literature, I am really intrigued and amazed to see the huge variety of things that people are now doing with the leaf wax components. So I think it was a worthwhile expedition, and it certainly was great fun while it lasted. So anyway, all the best for the conference, and I'll hand you over to the next speaker.